So now the sixth study I was hoping that we could talk about was by Arntzen and Precht, and it was published in 1995. And so for this particular study, the authors measured coral reefs, and they were looking specifically at both the evenness and the richness of coral reefs. So again, we've mostly been talking about richness, but evenness again is just that kind of measure of the relative abundance of each species. And they were looking in Belize at some barrier reefs at these corals. And so at each site, they also were able to estimate, in addition to diversity, a measure of disturbance. And historic disturbances are rarely recorded in coral reefs, and so what they did instead was they used a proxy, and specifically their proxy was topographic complexity, which you can see here on the bottom axis. And so they assumed that disturbances like hurricanes that may come along at varying times would actually go in and break up all the rough water that would come in, would go and break up some of these fragile kind of branched corals. And so then clearly after these disturbances, the corals would kind of build back up and recolonize, and they would have more intricate kind of branch shapes. So clearly this kind of topographic complexity then represents our measure of disturbance. And so in essence, the more complex you have, the less disturbed the site. So if it's not very complex, that means it just got hammered and got all the things broken off, and so it's really not complex, it's highly disturbed. And so again, we see some really interesting patterns, okay? In the regions where you actually have low disturbance, which is kind of down, or pardon me, rather, it's up in this area where we have high topographic complexity, we can see that we have low evenness and low richness. That's also similar to when we actually have very, very high disturbances in this region, and again, we also have low evenness and low richness. Okay? And those are for different reasons, because in this region where you have very, very disturbed sites, in some cases it's so disturbed that nothing can establish, and it really kind of tamps down diversity. At the opposite end, where we don't have much disturbance, it actually tends to be a pattern that is really promoting the fact that there's not much, there's a lot of availability of, there's not much disturbance, not things going on, and so just a few species are going to predominate, and they're going to take over and really dominate over other individuals and keep richness down, and in this case evenness as well. And so there's this real sweet spot right in the middle at these kind of intermediate disturbances where you actually get the greatest species diversity. And so that's kind of our intermediate disturbance, this idea that in this particular region, that's where you have this greater diversity. And this is something that students often forget about, that you can have these nonlinear shapes such that it does not always highest at one end and lowest at the other. But in this particular case, as we look at this axis of disturbance, that it's at the intermediate levels where disturbance is the greatest. So high and low are not necessarily good. It's just in the middle that's just right. Okay, so this next, the seventh example data set that I wanted to talk about was published by Stevens and colleagues in 2006. And so for this particular study, the authors surveyed grasslands across Great Britain. And so at each site, what they did is they measured plant species diversity as well as nitrogen deposition. And so nitrogen deposition can be caused by many things. It can be caused by volatilizing animal waste, synthetic fertilizers, burning of fossil fuels, lots and lots of different factors. But if they went to a whole bunch of different sites, then they could get a range of deposition rates. And you can see there's two different axes, this NHX or NOx, and those are just different forms of nitrogen. So it doesn't really matter. We just know that in this case, that in this end, we actually have lots of nitrogen, this end, lots of nitrogen, in essence, really, really high nutrients. And as we can see, if we do indeed look at the plant diversity in relation to nitrogen, we see this nice, clear, negative linear relationship, meaning when they are really, really nutrient rich, you actually have lower species, okay? And that is something that, again, is not as intuitive. Having low nutrients promotes greater diversity. Students tend to think about in their house that they give their house plants some nutrients and it makes them grow. But that's just one individual species. If you're talking about an entire community and looking at a diversity, you're actually better to have lower nutrients because it allows for a kind of availability of different ecosystems and different niches that can promote the growth of different species. Whereas if you flood the entire area and it becomes all nutrient rich, again, you tend to have this one particular species or maybe just a few that take over, and they just take over all of the space, take over all of the nutrients, and they outcompete any other species there. And so then for our final data set, it was published by Kerr and colleagues in 2001. And so for this particular study, the authors actually used remotely sensed data using satellite imagery of land cover. And the imagery detects different types of vegetations at a really, really large scale. So they actually created a grid to cover all of Canada with these kind of grid cells that were about the size of 5 degrees, or if you're not kind of knowing about that, it's about 550 kilometers along one side. So they're really, really big cells, but they wanted to cover all of Canada. And so within each grid, they actually calculated how many different habitats there were. And so you can see along this bottom axis, that was that value. And so clearly in these particular regions, these are these grid cells that are really, really heterogeneous. They have lots and lots of different habitats within one cell. In contrast, if you were to look down here, that's one cell that's covered by maybe just 1 or 2 different colors, or in this case, I guess it would be about 6. So it's really not, it's more homogeneous, if you will. And they also were able to get some estimates in this same region of the diversity of butterflies using data that was actually collected and accumulated in the Canadian National Collection records. And so when they looked at these two pieces together, there's clearly a nice correlation, a positive linear correlation, that as you have greater heterogeneity on the landscape, you actually can promote butterfly diversity. And that's a pretty classic pattern, that as you have more of this heterogeneity, you do increase diversity. And so those are all of our data sets. And clearly what we've been doing is trying to look at the factors that promote diversity. But clearly we can go back to each of these and identify what are the factors that reduce diversity. And there's many, many different things. So one thing is, as we just mentioned, if you have less habitat heterogeneity, if it's more homogeneous, then there's fewer niches, and there's not as many places for different unique species to come and colonize. Another factor is if there's more nutrients. So again, think of that nutrient hog that comes in and competitively excludes everything. It just takes over, and there's not a whole lot of room for other species to colonize, which keeps diversity down. Another thing is to look at diversity, or look at disturbance rather. And again, we need to remember that there's this intermediate sweet spot. So really frequent or really intense disturbances are not good because they don't allow anything to establish. But at the other end, if you have things where there's very infrequent or very minimal disturbances, we just have these really competitive species, just like we saw with the nutrient case. In addition, if you have higher density, that's again not such a great thing because there's a whole lot of things competing for those same resources. There's just less space for each individual. And we can go on, because we can say the smaller the species pool, that's also not a great idea. So if you don't start out with many species to be able to go around or colonize an area, then clearly it's not going to become diverse in the end. This classic case is with islands. If you have an island way out in the middle of the ocean, it's not going to have a whole lot of diversity as compared to one that's closer to the mainland, which actually has a greater potential influx of more species. Another thing is area size. The example that we saw the data set was about looking at lakes, but you can clearly do the same thing with islands or maybe even mountaintops. The smaller the area you have, the fewer resources and the fewer space and the less diversity that it could potentially have. General patterns in general tend to be that higher latitude and elevation also have lower diversity, and this usually is correlated with having cooler temperatures and less moisture. And then finally, having these species interactions can be a good but can be a bad thing. Clearly it's nice to have them because they kind of help tamp down or kind of hold at bay some of those really competitive species. But if you have too much of it, clearly it's a heavy disturbance, and it's really not going to be a positive thing. And so clearly there's lots and lots of factors that can increase diversity and decrease diversity. I would advise that you kind of make a list for yourself so that you can use that to study from.